This is the scientific process module. We are part of the Introductory 1 biology team. These are the learning objectives that we covered in the module. We were interested in having students understand the parts of the scientific process. Students needed to be able to define a hypothesis and identify the qualities of good and bad hypothesis. They needed to be able to distinguish between observations, hypotheses, and predictions, make their own testable hypotheses, and understand experimental design. Given a data set, students need to be able to understand and identify the independent and dependent variables and describe the relationship between them. They also need to identify and create the appropriate type of graph for a given data set and describe that graph using quantitative statements and differentiating between linear and nonlinear relationships. Finally, they need to be able to draw appropriate conclusions from data and evaluate the validity of those conclusions. Here are the quantitative competencies covered in the module. Competency 2, students need to be able to interpret data sets and create an appropriate graph to visualize the data. Competency 4, students must be able to identify and compare nonlinear relationships, and while looking at a graph, be able to identify the independent and dependent variables and how they are related to one another. Competency 5, students need to be able to define a scientific hypothesis and understand experimental design. They need to be able to distinguish correlation and cause and be able to evaluate whether scientific conclusions are warranted. Competency 6, students have to analyze graphs and use quantitative language to describe them.
The target student population. These are first-year introductory biology students with varying math skills. All the participating institutions have different math requirements for this course, and so we designed the module to fit the needs of such a diverse population. The module is made up of 3 parts. The first is a preparatory assignment that they complete at home. The second is the in-class component that consists of 5 activities. The third is the summative assessment that's given at the end. And the time it took to complete the in-class assignment was approximately 2 hours. And both HCC and MC did this in a lab setting. So this is designed to be completed early in the semester. The preparatory assignment or the pre-work was completed at home, and they turned it in. And these are some examples of the pre-work. Parts 1 and 2 allow the students to go through the steps of the scientific process through matching activities, and they reviewed the vocabulary terms. In part 3, students looked at different types of graphs, and they also determined correlations in the examples between the variables in the examples that were given. The fourth part of the pre-work was an introduction to making a graph, where the students went over the components of a graph, and they also identified errors in pre-constructed graphs like the ones you're seeing on the screen. And they pointed out what was missing or what was wrong. Like the title may be missing, or the scale is off, or maybe the axis is not labeled. So once they completed the pre-work, the in-class assignment consisted of 5 activities, and the students completed the worksheets in groups of 2 or 3 and discussed their answers. Activity 1, this is where they looked at whether a particular hypothesis is a good or a bad one, and if possible, to rewrite the bad statement to make it a good hypothesis. Activity 2, the students identified the independent and dependent variables in a set of examples and determined whether these variables would be placed on the X or the Y axis of a graph. In activity 3, the students focused on identifying the control and the experimental groups in the given examples, and then the students also interpreted the results of an experiment that were represented on a graph. The fourth activity had the students determine the positive and the negative controls after they had identified the control in the experimental groups. They looked at the positive and the negative controls in an experimental setup, and they understood the importance of each type of control.
In the final activity, activity 5, students put together all of the concepts that they learned from the pre-work and other activities. They are given a scenario. Industry scientists are testing a new active ingredient in sunscreen to see if it is more effective than a sunscreen that is currently in use. The scientists used 3 treatments on the test subjects: lotion with no active ingredient, lotion with the active ingredient that is known to be effective, and lotion with a new active ingredient. So they had a negative control, a positive control, and an experimental treatment. Then scientists measured the development of redness in units per square inch every 5 minutes for a total of 30 minutes. Students are then given a data table. With this scenario in mind, students used the skills they built throughout the module to identify the question, write a hypothesis, state a prediction, identify the independent and dependent variables, and all of the controlled variables in the experiment. They have to identify which treatment of lotion is the experimental treatment and which is the positive and negative control. Then, using the data, students construct a graph that best visualizes the data and draw conclusions about the effectiveness of the new sunscreen versus the one currently in use. This is an example of the graph that a student would make. They would construct a multiple line graph to best visualize the data. So they're looking at the development of redness over time. The graph needs to be properly labeled and drawn to scale, and they would conclude that the new sunscreen, which is the green line, the Sun X, is more effective than the one currently in use, which is the red line, the Sun-Kissed. We used one summative assessment question at the end of the module that tested these learning objectives. Describe a graph using quantitative statements, differentiating between linear and nonlinear relationships, and draw conclusions from experimental data and evaluate the validity of those conclusions. The question deals with the zombie apocalypse. Zombie hunters want to find out if temperature affects the number of zombie attacks. So they measure the average temperature and number of zombie attacks over a 30-day period. The question asks, which of the following statements is or are supported by the data? More than one choice can be correct. A, there is a positive correlation between temperature and zombie attacks. This is the correct answer. B, there's a negative correlation. C, no relationship. D, higher temperatures cause an increase in zombie attacks. This tests if students understand the difference between correlation and cause. And E, zombie attacks increase ambient temperature. The results of the summative assessment were surprising. It was consistent between both colleges, Montgomery College and Howard Community College. If you look at the top row, almost no one answered the summative assessment completely correctly. All students seemed to understand that zombie attacks and temperature were positively correlated. That's the second row. But almost all students guessed that temperature caused an increase in zombie attacks, the third row. So this was disappointing because students didn't seem to understand that correlation is not cause. We learned some important lessons for next time. We noticed that the pilot labs varied quite a bit in the kind of introductory lectures that were given by the faculty. To help with this, we agree that we need to provide more thorough written introductions to each module to explain the relevant concepts and vocabulary, just in case students are not exposed to it in lecture beforehand. The results of the summative assessment showed also that students did not learn the concept that correlation is not cause. The pre-work discusses correlation and cause and directs them to helpful websites, but does not have students answer questions about it directly. So we decided to add correlation cause questions to the pre-work and add an explanation of it into the intro section. In activity 5, we noticed that a lot of students were confused about whether to draw a bar graph or a multiple line graph. Students also had trouble understanding that treatment group was the independent variable in the sunscreen experiment. Based on a recommendation from someone in the larger intro bio group, we decided to have students construct a bar graph first and use total redness after 30 minutes as the dependent variable. After constructing the bar graph, then students would complete the multiple line graph to visualize the rate, the change in redness over time. This will help them understand when to use a bar graph, and it will also help them understand that categorical data can also be used as the treatment group can also be used as an independent variable. We also plan to add a multiple line graph question to the pre-work so that students have been exposed to them, to seeing them before they come to class. So these are the people who contributed to our module. Thank you.